Rio Grande River is one of Albuquerque's most beautiful features.
According to measurements from today, the water flow is about as weak as it's ever been over the past 50 years.
Scientists working along the Rio Grande recently made a discovery so alarming that it prompted an immediate red alert across multiple research and governmental agencies. The event began with an expedition to monitor the river's changing chemical and ecological makeup, a routine operation that has grown more urgent in recent years due to worsening droughts and increased industrial activity along the riverbanks. But this time, what researchers encountered in the water changed everything. Buried in the sediment near a remote bend in the river was a previously unknown microorganism, a biological entity with properties that defied current scientific understanding. The organism, microscopic in size but potent in nature, exhibited a previously undocumented capability. It could rapidly alter its environment, breaking down organic compounds at unprecedented rates. And that wasn't the only surprise. Before we start, smash the like and subscribe buttons for more updates. The microorganism demonstrated behaviors reminiscent of synthetic biology, as if it had been engineered rather than evolved. Genetic sequencing revealed strands of DNA that did not correspond to any known life form on Earth. These sequences carried elements of known bacterial and viral genes, but in configurations never seen before, raising the possibility that this organism may not have originated through natural evolution. Its genome included highly complex code segments arranged with a precision resembling digital encoding. Some sequences were mathematically consistent with Fibonacci patterns, a trait sometimes found in nature but rarely in microbial genetics, and never to this degree. This suggested either a previously undiscovered natural principle or something far more unsettling. Equally troubling was the microorganism's astonishing ability to replicate. In controlled environments, it could double its population in under 30 minutes, adapting its metabolism to whatever medium it was introduced into: river sediment, contaminated water, even nutrient-depleted solutions. Unlike most organisms that require specific environmental conditions to thrive, this one exhibited a chilling flexibility, surviving in extreme pH ranges, high salinity, and even low oxygen environments. The implications were immediate and sobering. If this microbe escaped its controlled environment and spread, it could destabilize entire ecosystems, starting with the already vulnerable Rio Grande watershed. Further analysis revealed that the organism secreted a substance with highly reactive chemical properties. This excretion, initially thought to be a waste product, actually accelerated the breakdown of synthetic pollutants, including microplastics, hydrocarbons, and heavy metals. At first glance, this seemed like a potential breakthrough, a biological tool that could help decontaminate polluted waterways. But the reality was far more complex and dangerous. In breaking down pollutants, the organism released byproducts that included previously unseen molecular configurations, some of which appeared to interact with surrounding flora and fauna in unpredictable ways. Fish exposed to the substance exhibited rapid mutations, including abnormal growths and behavioral changes, while native plants began to deteriorate at an accelerated rate. As word of the discovery spread through scientific circles, a flurry of international attention descended on the research team. Independent labs confirmed the findings, and calls for immediate containment procedures began echoing across the environmental research community. One of the most alarming aspects was the microbe's interaction with biofilms in the river, those thin layers of microbial life that coat submerged surfaces. The organism didn't just integrate with the biofilms, it dominated them, outcompeting native microbes and essentially rewriting the microbial language of the ecosystem. This kind of microbial imperialism had never been observed in such a rapid and aggressive form, and it left scientists scrambling to understand the implications for food chains, nutrient cycling, and river health. The microbial invasion also raised questions about its origin. Some speculated that it could be an unintended byproduct of industrial dumping, the result of synthetic biology experiments gone wrong and flushed into the river through poorly regulated waste systems. Others leaned toward the theory of a naturally occurring extremophile that had remained hidden in geological niches until recent shifts in river temperature and chemistry created a suitable habitat for its emergence. There were even a few, though cautious, voices suggesting something more speculative, that the microorganism's genetic structure hinted at directed design, perhaps from a lab, perhaps even extraterrestrial in origin, though such theories remained at the fringe. Regardless of where it came from, the consequences were immediate. The Rio Grande, already under pressure from agricultural runoff, shrinking flow rates, and habitat destruction, now faced an invisible invader with the capacity to fundamentally rewrite its biological architecture. Water samples from downstream showed trace amounts of the organism's byproducts, and while not yet catastrophic, the trend pointed toward a rapid spread. Ecologists warned that if the organism reached certain wetland regions or tributaries with higher biodiversity, the resulting ecological upheaval could be irreversible. The situation demanded action not in months or weeks, but in days. Environmental agencies responded by launching a multi-pronged investigation and containment operation. But the organism had already demonstrated an alarming capacity for dispersion. It hitchhiked on fish, latched onto boats, and even showed resilience in airborne water droplets during high winds. The fear was not just that it could spread throughout the Rio Grande, but that it could leap to other watersheds via human activity or migratory species. The potential for this microbe to jump ecosystems opened the door to a scenario where water systems across the continent could be silently overtaken before the full scope of its behavior was understood. Even more disturbing was the organism's effect on human infrastructure. Pipes and filtration systems that drew water from affected regions began showing signs of corrosion far beyond what would be expected from standard pollutants. Materials like PVC, copper, and even treated steel degraded in record time when exposed to water samples containing the microorganism. Preliminary laboratory models suggested that the excretion produced by the organism could catalyze reactions that undermine the molecular integrity of common industrial materials. If this proved true on a broader scale, the implications would extend far beyond environmental collapse to potential failures in water treatment facilities, irrigation networks, and municipal plumbing systems. Adding to the complexity of the situation were the ethical and scientific dilemmas presented by the organism itself. On one hand, its pollution-destroying capabilities were unrivaled. If harnessed safely, it could revolutionize waste treatment and environmental restoration. On the other hand, its volatility, aggression, and unpredictable byproducts made it a biohazard of the highest order. Debates flared within the scientific community about whether to destroy all samples and erase its existence, or to preserve it for further study in the hope of unlocking its potential. These conversations were not academic. They involved direct risk calculations, weighing the future of ecosystems against the tantalizing possibility of a technological breakthrough. As researchers pushed their analysis further, they discovered a unique protein structure within the microorganism that acted as a kind of switch, regulating its behavior based on environmental stimuli. This suggested a level of sophistication not typically seen in natural extremophiles, and it intensified suspicions about artificial origin. The protein complex responded to light, salinity, and even electromagnetic fields. It could upregulate or downregulate the organism's reproduction rate, excretion levels, and metabolic pathways. In essence, the microorganism seemed capable of sensing and adapting to its surroundings with an efficiency that rivaled digital systems. If this switch could be manipulated, scientists argued, it might be possible to control the organism's behavior, to turn it from threat to tool. But others cautioned that any attempt to engineer such control mechanisms risked triggering unintended mutations or escape scenarios. Monitoring stations along the Rio Grande were hastily upgraded, with sensors designed to detect trace chemical signatures from the organism's excretion. Meanwhile, drone teams were deployed to scan remote riverbanks for signs of biological disruption. In some regions, telltale indicators began to appear: water turning unusually clear, algae blooms disappearing, insect populations shifting dramatically. While this might sound like an improvement, ecologists knew better. Rivers operate as finely balanced systems, and sudden clarity often means microbial collapse. What followed was a cascade of changes up and down the food chain. Species reliant on algae for sustenance vanished, while opportunistic invaders began to rise. The river's identity was being rewritten, and the script was coming from an organism that no one fully understood. The potential for zoonotic crossover added yet another layer of concern. Could animals exposed to the microbe become carriers? Could it affect livestock drinking from the river? Studies began on mammals exposed in controlled settings, and early results were unsettling. Lab mice that ingested water containing trace amounts of the organism showed gastrointestinal changes and alterations in gut microbiota, some of which persisted for weeks. The possibility that the microorganism or its byproducts could subtly reprogram digestive ecosystems in animals, or even humans, raised red flags with virologists and epidemiologists alike. The specter of a new class of biological disruptor loomed, one not defined by direct infection, but by indirect ecological and metabolic sabotage. Geopolitical implications began to surface as well. The Rio Grande forms a natural border between the United States and Mexico, and both nations now found themselves staring at a shared biological threat. Coordinated efforts were complicated by bureaucratic hurdles, differing scientific protocols, and jurisdictional overlaps. Still, urgency had a way of cutting through red tape. Bi-national task forces were assembled, and secure research labs across both countries were mobilized. Quiet diplomatic conversations began about worst-case scenarios, how to handle a full-scale river collapse, how to protect downstream populations, and how to respond if the microorganism made the leap into agricultural or urban water systems. At the core of all this was a singular truth: the rules had changed. The discovery in the Rio Grande was not just another environmental anomaly. It was a signpost, a flashing warning that something unprecedented had entered the biosphere. Whether it was an accidental release, a natural wonder, or something far stranger, the microorganism represented a disruption to the delicate equilibrium that sustains ecosystems, economies, and human health. Its power to degrade pollutants and destabilize food chains simultaneously created a paradox: a savior and a destroyer wrapped into one microscopic enigma. As night fell on the research camps along the riverbanks, with floodlights casting shimmering reflections on the water's surface, a new kind of vigilance took hold. This wasn't just about protecting a river anymore. It was about understanding what happens when the boundaries of biology and technology, of nature and creation, blur in a place as vital and vulnerable as the Rio Grande. The red alert issued by the scientific community wasn't just a call to action, it was a plea to listen, to learn, and to act before the organism that lay hidden in the current became the catalyst for an ecological reckoning. The initial shock of the discovery along the Rio Grande rippled outward, but as deeper investigation unfolded, scientists began to realize that the microbe's implications extended far beyond immediate environmental hazards. The organism's biochemistry exhibited properties that blurred the line between biological and synthetic systems, introducing a new frontier of questions about what life could be and how it might operate. The Rio Grande, long a symbol of natural boundaries and historical divides, had unexpectedly become the stage for a scientific paradigm shift. One of the most striking revelations was the microorganism's energy metabolism. Unlike conventional life forms that rely on glycolysis or photosynthesis, this microbe used a redox process involving rare earth elements, notably lanthanum and cerium, extracted from the sediment. This biochemical anomaly meant it was not only independent of traditional nutrient chains, but also capable of surviving in nutrient-void environments by tapping into trace minerals. When researchers tried to replicate its activity in laboratory settings, they encountered a previously undocumented process of chelation. The organism could draw and bind these metals from virtually inert matter. In doing so, it initiated chemical chains that resembled primitive battery systems, generating voltage differences across its cellular membranes far exceeding anything typical in microbial species. This suggested the potential for bioelectrical capabilities previously thought impossible in such organisms. These findings introduced a disturbing scenario. If the microbe could operate in energy-deprived environments and convert raw minerals into metabolic fuel, there was no known natural boundary that could confine it. It would be equally at home in arid deserts, urban sewage systems, or the deep sea. The idea of containment grew increasingly tenuous as evidence mounted that the organism was not bound by typical biological limits. Even more concerning was its ability to enter a cryptobiotic state, a form of hibernation where all metabolic activity ceased, allowing it to survive desiccation, extreme heat, and possibly even UV radiation. If it could persist in this state and later reactivate under the right conditions, its dispersal might already be far more widespread than anyone imagined. Surveillance of birds and insects around the river revealed another unsettling development. Traces of the microorganism were found in the digestive tracts of certain bird species that fed along the banks. These animals, capable of long-distance migration, posed a potential vector of continental distribution. Analysis suggested that while the microbe did not immediately harm these animals, it formed a passive symbiosis in the gut, residing within without triggering a full immune response. The possibility that the microbe had developed or evolved strategies to evade detection by host immune systems sent another shockwave through the medical community. Such stealthy integration hinted at an evolutionary trajectory that was either vastly accelerated or artificially influenced. Meanwhile, teams examining the riverbed geology uncovered something even more enigmatic. Microscopic biofilms dating back several decades, possibly centuries, containing genetic traces of what appeared to be earlier, less complex iterations of the organism. This sparked a new hypothesis. The microbe hadn't just appeared recently, but had been evolving in hidden isolation, perhaps in subterranean aquifers or deep sedimentary layers shielded from modern influences. If so, recent shifts in temperature, hydrology, or chemical pollution may have disturbed its dormant life cycle, catalyzing its emergence into the broader ecosystem. In this light, the red alert wasn't just about a new arrival. It was about a long silent entity waking up. Speculation turned to the potential evolutionary implications. Could this organism represent an entirely separate branch of life? One that had developed in parallel, hidden from mainstream biology? Some proposed a radical classification, not a bacterium or archaea, but a new domain of life altogether. Its cellular membranes, ribosomal structures, and enzymatic pathways were unlike anything cataloged in current phylogenetic trees. This shook the foundation of evolutionary theory, forcing reconsideration of the notion that all terrestrial life shared a common origin. If life could evolve independently in such a radically different way on Earth, it widened the window for extraterrestrial life forms to exist, not just on distant planets, but in forms we may have overlooked entirely on our own. Amid these revelations, attention turned towards synthetic biology and advanced AI. Could the organism have been a result of artificial design? Several features strongly resembled modular genetic engineering. For instance, its gene clusters were arranged in operons that resembled the plug-and-play format used in synthetic DNA assemblies. These units could be expressed or silenced based on external triggers, almost like a programmable organism. Additionally, its ability to process and adapt to environmental data bore hallmarks of machine learning-like behavior. The microorganism seemed to learn from its surroundings over time, optimizing its chemical outputs and adjusting its behavior in repeated trials within laboratory setups. This fed into fears that someone, somewhere, had once engineered a living system capable of iterative learning, and that it had escaped or been lost, only now resurfacing in the Rio Grande. The implications for security and defense were stark. Governments began to treat the incident not just as an environmental emergency, but as a potential biosecurity threat. Intelligence agencies discreetly launched inquiries into whether any classified projects could be connected to such an organism, while defense analysts began to simulate scenarios involving its deliberate use. If engineered and weaponized, the microorganism could be deployed to disrupt enemy infrastructure, corroding water systems, crippling agriculture, or even altering populations of native species in a target ecosystem. Its resilience and capacity for adaptation made it uniquely difficult to contain, neutralize, or even detect in time to stop widespread damage. However, not all visions of the organism's future were apocalyptic. Visionaries in biotech circles began to envision new applications. If the microbe's breakdown of pollutants could be tamed, it might revolutionize waste treatment, converting toxic waste into inert or even useful byproducts. If its cryptobiotic capabilities could be isolated, It might pave the way for medical innovations such as long-term cell storage or new forms of drug delivery. Even its unusual protein structures held promise for nanotechnology, possibly offering a blueprint for self-assembling materials. But every potential benefit was tangled in layers of risk, and no one could yet claim to fully understand the cost of meddling further. As more data poured in, the microbe's presence began influencing human behavior along the river. Communities that relied on the Rio Grande for agriculture and daily water needs grew increasingly anxious. Farmers began noticing strange growth patterns in crops irrigated with river water. In some cases, plants grew faster but exhibited abnormal structures, such as translucent leaves or unexpected pigmentation. Soil samples taken from fields bordering the river showed shifts in microbial composition, with native bacterial populations giving way to unknown strains that appeared to be genetically related to the river organism. These changes weren't always destructive. In fact, some plants showed improved drought resistance, but the unpredictability made the situation untenable. The social consequences soon followed. Rumors spread faster than scientific facts. In towns along the river, whispers of contamination, mutations, and invisible sickness created a fog of fear. People began avoiding the water entirely, and water trucks had to be brought in from uncontaminated sources to meet daily needs. Fishing industries collapsed, not because of documented toxicity, but because no one wanted to eat a fish that might be altered in unknown ways. Tensions flared between communities on either side of the border, each fearing the other's response or perceived inaction. Trust in institutions faltered as the narrative spiraled beyond what officials could control. If you like this video, please give it a like and subscribe to our channel. Also, leave your comments below and tell us, what are your thoughts on scientists' Rio Grande findings? Will it impact us immediately? We want to hear from you. Thank you for watching, and see you next time.